My name is Delara Motlagh. I am the Chief Operating Officer at Fujifilm Cellular Dynamics. So I look after the business holistically. All of the functions report to me, including operations, manufacturing, and development. iPSCs have held a lot of promise for many years. And so if you look at the different stages of development from discovery through manufacturing and scalability, there are a few key challenges that must be tackled early on. First and foremost, iPSCs are a cell source. They are not the final therapeutic material. So one of the early things that must happen is determining what is the cell type that you're differentiating into and establishing the process that can efficiently generate this. This is one of the first hurdles, and not only scientifically in the differentiation process, but in developing that process such that you have the end in mind and you are having something scalable from the beginning. Next is going into manufacturing. So once you move into the manufacturing environment, you have a reproducible process that you have defined. Now it's putting it into a standardized process that can be repeatable and through various different hands. And this is where the translation of the science hits the engineering. This also can be tackled early on by looking at your key process parameters and ensuring that not only do you understand what it takes to get to your final cell type, but to also ensure that you have the proper controls in place such that your manufacturing can be managed step by step. And so these are some of the key learnings in order to be able to harness the power of these cells. So when you think about scalability for iPSCs, you have to really consider it in two parts. There's the scaling of the actual iPSC cell banks in order to have that raw material or source material for your cell therapy. And then there's the scaling of the final cell type. Depending on what that cell type is, your scaling strategy may be to make a large number of iPSC banks and then do the differentiation so you minimize the scale of the final cell. Or you may take the opposite approach where you will scale to a smaller extent the banks, but then those banks, those iPSC banks, can be differentiated and then that cell product can be scaled. And I make that distinction because these are very different cell types. They will also behave differently. So for example, you may have an adherent population. iPSCs are adherent cells. However, depending on what you differentiate them to, they may be either suspension or adherent. And so the scaling strategy will look different. So these are key considerations, as well as ensuring stability of the cell, not only in the line, but also in the final cell type. And some cells are more amenable to multiple rounds of replication and scale, and others are not. So these are key considerations to take into account. And then ultimately, as you're scaling your process, it's not just about the final number, it's about the reproducibility from each step of that process. So when you think about process development and design principles for iPSCs, it's always important to think of the end state that you want to be. So I always think of this as have the end in mind. And one very important grounding principle is quality by design. At the beginning of this process, when you know what it is you're trying to make, really defining what that end state for the product looks like early is really important. What does that cell look like? How do you need it to behave? What are the critical process parameters to get there? And the final attributes of that cell. What's its phenotype? What is your expected viability? And so on. Defining these early gives you a roadmap to be able to do the process development thoughtfully. Otherwise, you're always in a reactive state, and it's very difficult to stabilize the process at that point. Platform development is key for cell therapy overall. and in addition to that for IPS. The platforms themselves will not solve the problem. The platforms are a tool to help you automate and to potentially scale your process. So you must first have a process that's ready for that automation and those platforms. And then it works hand in hand. Incorporating these platforms early is very beneficial because then as you're developing your process, this is incorporated in. The platforms themselves can vary tremendously on size and scale as well as performance. There are a number of different bioreactors, for example, that are more amenable to suspension cultures. From a high scale and throughput perspective, adherent cells like iPSCs, there are not large-scale adherent platforms today that can easily be used in the process. So this is an opportunity for all of our tool developers in the market. That being said, there are ways and workarounds to be able to incorporate them. So the key with the platforms is, one, understanding what you're asking of the cell type and how sensitive they are. Some of these platforms move the media in different ways that may have shear effects on the cells. Some are more sensitive than others. Understanding the dynamic of your cell type will be very important in choosing that platform, and then incorporating that in your development early allows you to save a lot of time, not only in the development, but once you hit clinical, to minimize the changes you need to make later. There are many gaps in the process as you try to bring therapies to market. And if you think of what are some of the barriers and hurdles early on, first, as you take an academic process, many of these processes are just that. They're fantastic science at a bench. And so they tend to be very open systems. They're really not designed to scale commercially. Taking those and making them less academic and to industrialize them is one of the key challenges. This can be done through platforms, but it can also be done through thoughtful design of the process. The second is an obvious one as we think of these are living therapies, and the ministries of health around the world have struggled with how do you regulate from the CMC packages to what is minimally required before you go into a human in a clinical trial. While a lot of progress has been made in this area, a lot of great collaboration with the scientists and the biotech community, there has also been some setbacks where we see a bit of inconsistency in the approaches around the world. Finally, as you think about making these therapies, these life-saving therapies more accessible, we have to think about access. Now, access comes in different ways. There's physical access, right? In which case, there's a lot of education that's needed of the physicians that are treating as well as the patients themselves. There's access in where can these be made available? Are they off the shelf? Do they require an intricate network? And so really thinking through how can to streamline the access for patients, and there's a lot of great work happening there. iPSCs really serve that purpose well as an off-the-shelf solution for the allogeneic, but of course needs to be worked into that accessibility. And then last but not least is the affordability. And so as you think of how to make them more affordable, there's some clear levers to pull in streamlining process such that the cost of goods can be reduced and therefore reduce the price tag. But I think even more fundamentally, it's understanding and articulating the value of these therapies and what it means for the healthcare system overall, so that they can be priced in a way that is affordable for the healthcare systems and the costs that they can take out of the healthcare system overall, but that value is preserved such that it continues to incentivize all the great innovation of bringing these therapies, not just to a few markets, but around the world. Manufacturing readiness and commercial readiness for therapies and iPSCs among them continue to evolve. Having a process be manufacturing ready means that it needs to be able to go into a GMP environment. So some of this starts with the selection of raw and source materials that can actually be used in GMP. This is a barrier that often has to be broached early in development when you're taking perhaps an academic process and trying to have it be ready for manufacturing. And then there's the development of the overarching process itself and ensuring that it can not only move into a GMP environment, but truly be able to produce at scale, both in number of cells, but also in the output. Do you have an appropriate number of vials? Do you have a process that can produce those vials in a compliant manner from a regulatory perspective as well as from a supply chain perspective? So it's really taking that commercial engine and baking it into how you're managing the process. And these are important considerations that for iPSCs in particular have become more accelerated, because as these programs go into clinic, they are enjoying more accelerated regulatory approval periods, such that they may go from an early human phase 1-2, straight into a pivotal trial. And so after you've done your pivotal, you don't want to be making a lot of changes. So really, that thinking of the commercial readiness happens in that phase 1-2. When you think of iPSC therapies, there are a number of different tools that are used, both in the reprogramming and the gene editing, as well as the differentiation. This innovation continues, and it becomes very important, particularly as you think about making them more accessible in various indications where you want them to be able to go into the body without having a reaction. So hypoimmune lines, for example, that can evade the immune system are very important, because otherwise you would need to suppress the immune system. And there are very elegant tools in gene editing that continue to evolve. And this is an area that has really helped to bolster the use of iPS as a cell source for therapies. And I'm quite optimistic about what this will do for their accessibility.